doing some really cutting edge work in nanomedicine, and so y'all would help me in welcoming Dr. Joy Wolfram.
Hi everyone, I'm very excited to be here, and I want to thank Ethan and everyone for inviting me here. And today I'm going to talk about synthetic nanoparticles and biological nanoparticles for cancer and beyond. So I'll start by talking about how we can exploit hemodynamics in nanomedicine, then how we can modulate innate immunity, and finally the use of extracellular vesicles, these biological nanoparticles. But before I go into each of these topics, I just want to give you a brief perspective on my view of nanotechnology. So as you know, it's technology on the nanoscale. So depending on the definition, 1 to 1,000 nanometers. And we know that viruses, for instance, are natural nanoparticles in this size range. And when we look at the impact of nanotechnology, it's affected many different fields. For instance, computer science, where these computer chips become smaller and smaller and there's great potential. Also solar power technology, where the individual units in solar panels become smaller. But then when we look at medicine, most conventional drugs are molecules, so less than 1 nanometer. And then a few decades ago, we saw a lot of antibodies reach the market, and they're around 10 nanometers. And today we also have at least 10 nanoparticles clinically approved for cancer in the US. But actually, interestingly, on the contrary to these first two examples, when we talk about nanotechnology in medicine, we're actually going larger than what we're used to. And so what are the advantages of going larger? Well, the first advantage is that if you have more space, you can add more function, so multifunctionality. The second advantage is that you get these unique electromagnetic properties that are not apparent on the molecular or macro scale. And finally, the third advantage that I believe nanomedicine has is that it's differently processed by the body, and we can really exploit this to change the final location of nanoparticles inside the body. And I'll briefly discuss all of these three advantages, but I'll start by talking about multifunctionality. And this is a simplified schematic of a multifunctional nanoparticle, just highlighting some of the extra functions that we can add through nanotechnology in medicine. The most simple example is solubilization. So we can take these chemotherapeutic agents that are not water soluble, that typically conventionally in patients are given with toxic solvents. But instead of using those toxic solvents that also have their own side effects, we can put it inside a nanoparticle or complex it with a nanoparticle. And a clinically approved example that I think most of you are familiar with is Abraxane. And Abraxane is approved for various cancers, and it's albumin, so protein-based nanoparticle. Another example of multifunctionality is protecting the cargo from degradation. And this is especially important when we deal with therapeutic molecules that are sensitive to breaking down, such as RNA. And so last year, it was a huge breakthrough for nanomedicine because we had the first small interfering RNA clinically approved called patisiran for an inherited disorder, and it's delivered in a lipid-based nanoparticle. Then we also can avoid immunorecognition by using nanomedicine, and an example of this is pegylation. So this polymer that hides the nanoparticle from the immune system by attracting a hydration layer. And several of the nanoparticles that are clinically approved are pegylated. Doxil is actually the first nanoparticle approved for cancer. We can add targeting ligands. Targeting ligands have traditionally failed in clinical trials. We're still trying to figure out the reason for this. But there are some in early phase clinical trials. We can add imaging agents together with the therapeutic agents to get a theranostic drug delivery system, and those are still mostly in preclinical studies. We can also add permeation agents that help break down the tissue. The extracellular matrix, for instance, they do have some side effects, so they're still not in the clinic. We can utilize various triggered activation strategies where a specific trigger in the microenvironment causes the drug to be released or the nanoparticle to be activated. And there are some examples of this, mostly probodies that are not traditionally considered nanomedicine, even though they're nano-sized. And finally, we can have various drug combinations because we have more space. And the advantage of this is we can get the optimal ratio of two drugs inside the same nanoparticle. And a clinically approved example is Vyxeos, which is again approved for cancer, and you have two different chemotherapeutic agents inside this liposomal carrier. And within my own research, I work with various multifunctional silicon particles, looking at strategies, for instance, for sequential drug release, and also worked with liposomes. So the next advantage of nanomedicine is these unique electromagnetic properties. And so they can be optical, magnetic, electronic, and thermal. And the take-home message is that they're not apparent on the molecular scale or macro scale, but only on the nanoscale. And the reasons for this is that nanoparticles have a large surface area to volume ratio that causes these unique properties to arise, but they're also in the same size range as the wavelength of light. And because of these features, we get unique electromagnetic properties. And something that we've done a few years back was to use gold nanoparticles for photothermal therapy for cancer. But there's also a clinically approved example of exploiting electromagnetic properties, which is a nanotherm, and it's clinically approved in Europe. And it is based on local injection of iron oxide nanoparticles, and it's actually approved for various forms of brain tumors. And the patient is then placed into this magnetic field, which causes the iron oxide nanoparticles to heat up and destroy the tumor through those thermal elevations. And they've been talking with the FDA for many, many years, so they're probably going to get approval here soon. The main difference with the European Medicinal Agency and the US is that the US wants anything that gets approved to be biodegradable. And these are actually not biodegradable. So they're found in patients' brains 5 or 10 years later, but that could actually be an advantage. If the tumor comes back, you don't have to do the injection again. You just place the patient in the magnetic field. Of course, this means that the tumor has to be in the same region. But I think they're going to get FDA approval shortly. And so the third advantage of nanomedicine is this differential processing by the body. And I'm going to spend a bit more time talking about this advantage because I think it's more complex than the other. And so when we talk about site-specific delivery of systemically administered drugs, so either through the intravenously through the blood or taken orally, we know that for conventional drugs, only 0.001 to 0.01% will reach the target. So these are conventional molecular drugs, and these numbers are very scary, especially if it's the first time that you hear them. So with nanoparticles, we have this tremendous improvement. With nanoparticles, around 1% of what we inject into the blood will actually reach where we want it to go. And this usually means that nanoparticles have improved therapeutic efficacy and reduced side effects because the delivery is more specific. But one of the questions we've asked is 1% enough? And how can we further improve site-specific delivery? And so within the field of drug delivery, we believe that treatment of disease is a problem of drug distribution. And ultimately, we would like to be as specific as possible to make as much of the drug go where we want it to go. And you know that all drugs have this therapeutic window, which is the range of drug concentrations. If you go below it, you don't see any efficacy, but if you go above it, you have all these side effects. But if we can find a way to specifically transport nanoparticles to therapeutic areas, then we can expand this therapeutic window. And here you see a simplified analogy of drug delivery, where the car is the drug delivery system and the road the biological environment. And in the middle, you see this fire pit that symbolizes the obstacles in the body that prevent the nanoparticle from going where it's supposed to. So in order to improve drug delivery, we can either upgrade the drug delivery system, and if it's really good, maybe can jump over this fire pit. And we have examples of doing this, and I'll talk a bit about that. But a less common strategy is actually to change the biological environment. And we've also done that by modulating the innate immune system. So I'll start by discussing these sophisticated drug delivery systems, which are exploiting hemodynamics in the tumor. And so what we always try to do when we design drug delivery systems is to take advantage of differences between healthy and pathological tissues. So one example is cancer blood vessels, and they differ in two distinct ways from normal vasculature. First of all, the blood flow is slower, and this is because when a tumor grows, it's a rapid process, and for the cells to survive, they really quickly need to recruit a vasculature network. And because this is such a rapid process, it's also disorganized. And this disorganization causes the blood flow not to be optimal, so you have certain regions within the tumor where it's flowing very slowly. The second difference are these fenestrations or holes in the vasculature wall. And again, this is because the vasculature is immature. So it doesn't have time to form this continuous barrier. So you end up with these vasculature fenestrations. And so we asked, can we take advantage of this? And the answer was yes. So we designed these disc-shaped microparticles that are around 1,000 nanometers. And these smaller nanoparticles to take advantage of the fenestrations. And I'll briefly talk about how each of these particles interact with the vasculature. So we know that this disc shape is optimal for flowing close to the vasculature wall, and this is because of fluid dynamics. And once it's flowing close to the endothelial cells, it has high chances to bind because of this surface area that is large due to the disc. So when we look at the cancer where the blood flow is slow, the microparticles will bind to the vessel wall, and they will actually, in most cases, permanently stay bound. But in healthy vasculature where the blood flow is faster, the microparticles will still bind, but they will be a transient binding and detach. And if we compare this to conventional small molecules, we know that small molecules are less affected by this shear rate and blood flow. So it's difficult to exploit this type of hemodynamics when we're only working with a molecular toolbox instead of on the nano size range. And this is just a schematic illustrating what I just mentioned. So in normal vasculature, spherical nanoparticles, which are actually the most common, all the clinically approved nanoparticles are spherical, all the ones in preclinical studies, most of them are spherically shaped, but they're not actually ideal for interacting with the blood vessel wall. They tend to flow more towards the center of the vessel. So both in normal vasculature and tumor vasculature, you don't really see this adhesion, while the discoidal microparticles, they marginate across the endothelium, and in the normal vasculature, it's a transient adhesion. The fast blood flow causes it to detach, while in tumor vasculature, you see more of a permanent adhesion. And so then, when it comes to the smaller nanoparticles, we know that they can enter tissue if there's holes or fenestrations in the vasculature. So in cancer, as I mentioned, there's these fenestrations. They can enter and interact with the cancer cells, while most healthy vasculature doesn't have these fenestrations, so they won't enter into the tissue while they're circulating in the blood. And again, if we compare this to conventional drugs, small molecules, they're so small that they will go everywhere in the body, regardless of whether that organ has vasculature with fenestrations. So again, very difficult to exploit this difference if we're only dealing with molecules. But on the nano size scale, we can take advantage of this. So now we have two differences that we've designed delivery vehicles to exploit. Why don't we combine those? So why don't we take the disc-shaped microparticle together with the nanoparticles? And so to do this, we've used porous silicon microparticles, and inside the pores, we've loaded smaller nanoparticles. And we call this the multistage vector because it has several stages. So after we inject it into the blood, the microparticle, the first stage, will bind to the vessel wall. Here you see the microparticle and these characteristic fenestrations in cancer blood vessels. Then the microparticle will start to slowly degrade because porous silicon is biodegradable, and as it degrades, it will release these nanoparticles that can then enter through the fenestrations. So that's the second stage. And then we have therapeutic agents inside the nanoparticles that can directly bind to the target, and that's the third stage. And so we've looked at this in many different models, mostly cancer and inflammation. But one example that I'll show you is melanoma lung metastasis. So the goal was to get high accumulation of this multi-stage drug delivery system in the lungs. And we compared the accumulation of liposomes, so conventional nanoparticle, to liposomes inside the discs. And we found that lung accumulation was substantially increased. Here in blue and black you see the multistage vector bars. The same when we compared it to polymeric nanoparticles, and polymeric nanoparticles inside the multistage vector, again we saw enhanced accumulation in the lungs. And so now we ask whether this improvement in site-specific delivery would translate into therapeutic efficacy. And there's many different groups here, but the last group is the multistage vector with therapeutic cargo, and you see that the tumor burden is decreased in this group in the mice. The survival is prolonged, and they have less metastatic nodules in the lungs. So clearly by optimizing the shape to take advantage of hemodynamics in tumors, we can also gain better therapeutic efficacy. And of course, my goal is always to benefit patients. That's why I do research. So what are we doing in regards to getting this to the clinic? So at the Houston Methodist Research Institute in Texas, I have a joint position. So at the 12th floor of this building, we have a GMP facility that can produce clinical grade particles. And the team there got a $60 million grant from the DoD to develop this technology into phase 1 clinical trials. It's led by my PhD advisor, Dr. Mauro Ferrari, and it's for breast cancer lung metastasis. So we're hopeful that we can get this into the first patients and that we can see potentially similar results as in our mice studies. So to conclude this first part of the presentation, there's several advantages of nanomedicine. We have multifunctionality because we have more space. We can add all these extra functions. We have these unique electromagnetic properties. And we also have differential processing by the body, for instance, slow blood flow in tumors and fenestration, so we can design this multi-stage disc-shaped drug delivery system. And this is the first way in which we can improve drug delivery is through nanoparticle design. But the second way is actually by modifying the microenvironment, which I'll talk about in the next part of my presentation. And so we looked at various ways to modify the microenvironment, but I'll talk about one of these, which is deactivation of liver macrophages. And so this is modulation of innate immunity. And so I mentioned that only 1% of nanoparticles that we inject into the blood will actually end up in the tumor. But what I didn't mention is that up to 99% will go to the liver and spleen, because the liver and spleen have macrophages that are designed to recognize nano-sized objects, like viruses and bacteria, but also nanoparticles. And so in a hypothetical example, what if we could block the liver and spleen, let's say by 1%? That's nothing. In the best case scenario, we could increase tumor accumulation by 100%. So this means that a small change in what goes to the liver and spleen could have major consequences for tumor accumulation. So can we find a macrophage switch? Can we find something to temporarily switch off the macrophages prior to injecting the nanoparticles? And so we did this through a drug repositioning approach where we screened various drugs, and we found that a drug that was already clinically approved, which is a fast way to help patients, is chloroquine, 70-year-old drug approved to treat malaria. It was actually the most effective at blocking these macrophages from internalizing nanoparticles. And so we looked at 3 different macrophage cell lines, including Kupffer cells, which are the specific macrophages in the liver. And we saw that in all cases, when we gave chloroquine, the nanoparticle uptake in the cell was reduced, and this was at doses that didn't kill the cells. So we're not killing the macrophages, but we're doing something to them that prevents them from taking up the nanoparticles. And we wanted to make sure that we were not doing the same to cancer cells, because if we were blocking cancer cells from taking up the nanoparticles, that would be counterproductive. And luckily, we found that in 3 different cancer cell lines, so this is breast cancer, pancreatic cancer, and lung cancer, chloroquine at these doses did not block nanoparticle uptake. So maybe something macrophage specific. And then before we went into animal studies, we wanted to make sure that we were giving mice a dose of chloroquine that was clinically relevant. So there's this FDA guidelines when you convert a dose from human studies to mouse studies or vice versa. And so we used that calculation of body surface area, and we actually ended up using a slightly lower dose than the standard human dose for malaria treatment converted into a mouse dose. And so the first thing we did was take healthy mice, and we looked at accumulation at different time points, and we wanted to see when these nanoparticles, in this case liposomes, accumulated in the liver to a larger extent than in the blood, and this was after 6 hours. And so we used this time point for our studies, and we also, as you always want to do, is to use a positive control in your studies. So in our case, it was an agent that had previously been described to basically kill all the macrophages. So very effective, but not clinically relevant, and it's called Clodrolip. So when you give it to mice after 24 hours, all the macrophages from the liver are gone. So when we gave this agent to the mice, we saw that we could substantially block liver accumulation, and plasma accumulation went up, which is what we wanted. But again, this agent we can't take into the clinic because we'll get a lot of side effects. Then when we treated with chloroquine, clinically relevant dose, we saw that there was a similar trend. The nanoparticles were more in the blood and less in the liver, so this was exciting. But could we potentially get higher tumor accumulation, because these were tumor-free mice? And so we used the breast cancer orthotopic mouse model of human triple-negative breast cancer. And as I mentioned, in our studies, only 0.7% of the nanoparticles that we injected into the blood were found in the tumor. So again, an indication of the barriers in the body that prevent site-specific delivery. When we gave our positive control that destroyed all the macrophages, we saw this tumor accumulation go up, and a similar trend with chloroquine. And you may think, this is not a lot, we're just doubling tumor accumulation, but sometimes that's the difference between life and death if you get double the amount of therapeutic agents to interact with cancer cells. And we wanted to make sure that this was not just the case for one type of nanoparticle delivery system. So we asked, could this be broadly applicable? And so we did the same with the discoidal microparticles. We pre-treated the mice with chloroquine. And again, we found that lung accumulation went up and liver accumulation went down. So potentially this innate immune system modulation could be used for many different types of nanoparticles. And we also wanted to make sure that we weren't killing the macrophages. So we saw that the positive control here in the middle, all the macrophages in green, were gone from the liver. But our chloroquine group, the macrophages look very similar to the control group. So we're not killing the macrophages, which would have side effects. We're just somehow switching them off. And then we tried to look at what is the mechanism by which chloroquine is blocking nanoparticle uptake in macrophages. So we did some proteomics, and we found a target that we verified with Western blot, which is PCALM. It's very unknown, some publications with Alzheimer's, but it's actually one of the clathrin-coded PITS proteins. So that's one of the ways that nanoparticles are internalized by cells is through clathrin-mediated endocytosis. But interestingly, this was the only protein that was suppressed compared to the other 2 proteins. So potentially this is one way by which chloroquine, this malarial agent, is blocking one of these proteins necessary for nanoparticle endocytosis in macrophages. And like I mentioned, we're always looking at how can we help patients. And in the US, you can give off-label use of drugs. So this could be given to any patient if the physician wanted today, combined with any of the clinically approved nanoparticles we have on the market. But in addition, we're also trying to do a clinical trial at Mayo Clinic to systematically look at whether chloroquine has similar effects in humans as we saw in mice, and that would be industry sponsored with one of the companies that has nanoparticles clinically approved on the market. So to summarize this second part of the presentation, this mononuclear phagocyte system is a major barrier in nanomedicine, but we can use pharmacological inactivation of macrophages to improve site-specific delivery by, for instance, using chloroquine that blocks one of these proteins involved in endocytosis. And so for the last part of my presentation, I'll talk about extracellular vesicles. And extracellular vesicles are lipid vesicles, so around 100 nanometers. And they are actually found and secreted by all cells, and they contain biologically active materials such as RNA and proteins. And they're important for cell communication, and so I actually call them the text messages of the cell. So if one cell wants to send a message to another one way to do it is through an extracellular vesicle, which can also protect that cargo, that message that is sensitive to degradation, for instance RNA, because there's a lot of RNases both in the blood and extracellular space that will immediately break down this RNA molecule. So actually to communicate through RNA, one way to do this is by encapsulating the matter inside an extracellular vesicle. And so the research question we've asked is, can we tap into the body's text message system to improve healthcare? Why do we want to do this now? Because there's a lot of recent evidence really showing that extracellular vesicles play important roles in basically all physiological and pathological processes. Why are we doing this in my lab? Because we've developed and optimized this cool isolation technique that seems to be very effective compared to conventional ways of obtaining these extracellular vesicles. And why is this important? Because if they're involved in all these biological processes, then imagine what diagnostic and therapeutic applications this could have, right? So if they're this important, then why are they not in the clinic at the moment? And I believe the main challenge has been production. How do we concentrate and purify these extracellular vesicles? The most common method is ultracentrifugation, but it results in low quantity. So you can spend 1 month around the clock doing ultracentrifugation to get enough to inject a single mouse. Low purity, you get a lot of contaminants, and that's not good if you're trying to ask a biological question, because is it really the extracellular vesicles that are doing something, or is it your contaminants that you're isolating? Like I said, very time consuming, labor intensive. There's a lot of manual pipetting steps, not clinically scalable. I think there's only one or two companies in the US that does ultracentrifugation clinical grade, but there's a 2-year waiting list and extremely expensive. And then it's like you in a roller coaster times a million. So there's going to be some damage. The vesicles are going to fuse, they're going to release their content, and potentially the membrane is going to get altered. There's very low batch-to-batch consistency, again, because you have the human error in the pipetting steps. And so we really wanted to provide and develop an alternative isolation method to overcome all of these challenges. And so what we've been using is tangential flow filtration. And here you see a video. And this equipment is commercially available, and it's actually advertised for protein purification. But if you specify the parameters and change the membranes and how you run it, you can actually use it for extracellular vesicles. And a lot of labs are now starting to use tangential flow filtration, but it really comes down to all the parameters, and the intellectual property and know-how is in all of that. And the reason we started working with this is that in Mayo Clinic, Minnesota, which is the main campus, for 15 years they've been doing clinical trials with measles viruses using this isolation method. And measles viruses, when you look at them under the microscope, they're identical to mammalian vesicles, same size, same shape, same membrane properties. So we didn't have to reinvent the wheel. We sort of optimized their viral isolation protocol that is used in the clinic for extracellular vesicles, and they go up to 50-liter batches, so it's very scalable. you can produce a lot of vesicles with this method. And how it's different from regular filtration is you push something through a membrane, you get a lot of clogging, but with tangential flow filtration, it's actually flowing in the horizontal direction, so you avoid a lot of that clogging. And once you get clogging, you're no longer separating according to the size you selected. And we do this in 2 steps. The first step, we use a 600 nanometer filter to get rid of everything that is above that size. And in the second step, we use a smaller filter to get rid of everything that is smaller than 30 nanometers. But at the same time as we're filtering, we're also diafiltering, so purifying, and we're concentrating. And then we characterize these vesicles using nanosite, and this microscope will tell us the concentration and size distribution of our vesicles. And so one of the first things we did is, okay, let's take the most common method, ultracentrifugation, and compare it to this new method that we've optimized to see what benefit we have. And we looked at the yield with ultracentrifugation versus tangential flow filtration, and we saw that we could substantially increase the amount of vesicles we were getting from the same starting material. And we also looked at protein contaminants. So here you have albumin, and with ultracentrifugation, as I mentioned, you have a lot of impurities, but with tangential flow filtration, we get rid of everything, or most of the protein that isn't nano-size, so smaller than 30 nanometers. And then we wanted to make sure that we can use this TFF-based method with various sources of materials. So can we isolate these extracellular vesicles from cell culture media? And we found that we could. And one thing to notice is that with ultracentrifugation, you have all these peaks all over the place, while the consistency is a lot higher with tangential flow filtration. And we made sure we had this extracellular vesicle enriched markers, and that we lacked calnexin, which is an intracellular marker. So anytime we do this, we want to make sure that we're actually isolating extracellular vesicles and not intracellular vesicles. But cell culture media is a very simple solution. So can we go to more complex solutions like fat that we aspirate from patients? And we found that we could, and again, we had good batch-to-batch consistency. We saw these nano-sized structures, and we had extracellular vesicle enriched markers, a little bit of intracellular markers here, but that's probably when the surgeon is doing this procedure, you're damaging some of the cells. So the 2 applications that we focused on now that we have this efficient method of obtaining the vesicles is the extracellular vesicle glycome for diagnostics, specifically for cancer metastasis, and secondly, adipose, so fat-derived extracellular vesicles for treating inflammatory conditions. And the one I'm going to show you today is myocarditis, which is heart inflammation. And so for the first project, we tried to find extracellular vesicle markers to predict which patients are going to develop metastasis. And if we know this, we can treat them with more aggressive therapy. And the problem is that when a patient does get brain metastasis, for instance, very few drugs, as you know, can cross the blood-brain barrier, so they don't have really a lot of options. And so we thought that we know that these cancer cell-derived vesicles are involved in the pre-metastatic niche. So a lot of papers a few years ago came out showing that the extracellular vesicles will be released from the primary tumor and then go to the site of metastasis to make it suitable for the cells to come at a later time point. And the interesting thing is that almost everyone is focusing on the RNA and protein content of these extracellular vesicles, while the glycome, the sugars, have been mostly ignored. And the glycome is very important for metastasis, and it's usually sugars that are on the surface of the extracellular vesicles, and as you know, the surface properties will dictate where they go inside the body. So we thought, can we look at the sugars on the extracellular vesicles, the glycans, to predict which patients will develop metastasis? So we teamed up with Dr. Borges at Arizona State University, and he has developed this glycan node analysis, which is an improved method for looking at glycans. And we used parental cell line, so breast cancer cell line, and we took it through several rounds in a mouse model to get a version that metastasized to the brain. So inject this human cell line into mice, let's say 20 mice, only one of those mice will develop brain mets. So you take those brain mets, you take them, put them in cell culture, you inject them into another batch of mice. Now 3 of those mice get brain mets, and you keep doing this until you get a cell line that is originally from the same cell line from a human, but now it goes primarily to the brain. And this is also published in Nature by another group, where you can see that 5 out of 5 of these mice develop brain mets. So now we're looking at how do these 2 different breast cancer cell lines differ in their extracellular vesicle characteristics when one goes 100% to the brain and the other does not. And so we started looking at the sugars between the regular extracellular vesicles and the brain extracellular vesicles, and we found some interesting things. For instance, this glycan node that we're looking into, whether it could be a biomarker for metastasis. But in addition to understanding if we can use it for diagnostics, we're also looking at the functional mechanistic role of these glycans associated with extracellular vesicles. And to do this, we've developed some simple cell culture assays where we see distinct differences in how these extracellular vesicles interact with brain blood vessel cells. So we see that they're taken up to a different extent. When we look at impedance, we see that they make the endothelial layer of these brain endothelial cells have different characteristics, and they actually cause differences in what gets from the blood side to the brain side in a in vitro cell culture model. So now that we're knocking down some of these glycans to see if we can see changes in these simple cell culture models. And the second project that we're working on is myocarditis. And myocarditis is inflammation of the heart, and it's actually responsible for 20% of all sudden deaths in the United States. And it's often due to a viral infection called Coxsackie virus B3. And there's currently no treatment for it, so we've teamed up with Dr. Fairweather, and she's developed a mouse model, one of the few in the world that closely resembles the human myocarditis condition. And it's a hybrid model between a viral infection and autoimmune model, because she takes heart proteins from the mouse and she mixes them with the virus and then injects it into the mice. And this way she has to use a very low viral load, which mimics the situation in humans. And so we see that when she injects the mice with this heart proteins and the virus, after approximately 10 to 14 days, you see this peak of inflammation in the heart that you can then look at through histology. So what we did is that we thought, can we use adipose tissue, so fat tissue, as an anti-inflammatory component for myocarditis? And this has been shown many times that if you take these mesenchymal stem cells, adipose stem cells, from the fat, their extracellular vesicles will actually suppress inflammation. And there's hundreds of articles on this, and there's various proteins and RNAs that are involved in this anti-inflammatory response. But unfortunately, there's some limitations of taking these mesenchymal stem cells. The first is that it's a very slow process. So once you take the cells or the fat from the patient, to actually get the stem cells takes at least 1 month, and you also need to expand them, which is virtually impossible to get enough material. It's very expensive, because if we're talking about doing this in patients, you then need to have those clinical grade facilities to expand these mesenchymal stem cells. The other problem is that it's not really a native environment. So it does work, but you're actually taking the cells out of their environment in the fat, and what you're doing is that you're putting them on a 2D environment. So what properties, what potential anti-inflammatory properties are we losing by doing this? And finally, you have to add a lot of exogenous agents such as cell culture media and so on. And as I mentioned, you have very low number of extracellular vesicles. So we thought, is it actually necessary to take this one cell population in the fat and get the extracellular vesicles to reduce inflammation? Or can we take the extracellular vesicles directly from the fat tissue? Could they have similar anti-inflammatory properties? And so this is what we do with our clinical collaborator, Dr. Shapiro, we take fat from patients subcutaneously from under the skin, and it's put into this FDA-approved device called lipogems. And lipogems, what it does, the starting product looks like this. It's usually not this bloody, but it will separate the cells from everything else that goes into this waste bag. And in the clinic, they typically inject the cells, for instance, into the knee of patients who have a knee pain. But this bag is thrown away all the time, and this bag contains, of course, all the extracellular vesicles and other things that is not cells. So we wrote an IRB to actually be able to characterize and use the waste coming from these patients. And side by side, we also take some of the cells from the same patient, but we take the bag, we run it through our tangential flow filtration to get the EVs. This takes a few hours compared to a month growing these cells in cell culture, making sure that they're mesenchymal stem cells, taking the cell culture media, then doing tangential flow filtration to get the extracellular vesicles. And so we've side by side compared these two populations of extracellular vesicles from the same patient. And we also make sure that when we're doing this, we can maintain sterility because this is important for clinical translation. But here you see that with only 50 milliliters of fat, so that's not a lot, we get enough to inject 50 mice when we take it directly from the tissue. So we get enough extracellular vesicles, which is really impossible to do through cell culture. And so we started comparing the extracellular vesicle characteristics. So this is a cryo-TM image of the extracellular vesicles from the cells above. So you see that there's extracellular vesicles. But when we looked at the fat, not only did we see extracellular vesicles, but we also saw structures like lipoproteins. And this is because our method of isolation is very specific for size-dependent isolation, but not for extracellular vesicles specifically. So we will get all types of nanoparticles that are present in the extracellular space of the fat. And this could also be an advantage, as we know that lipoproteins have many anti-inflammatory effects. So we've actually called them lipo-NPs, nanoparticles, because they're not just extracellular vesicles. But actually they have very similar size when we look at the cell culture-derived extracellular vesicles and the ones directly from the fat. But again, the amount we get is a lot higher. And so we compared the anti-inflammatory properties, specifically looking at macrophages, because macrophages are the ones that are driving myocarditis. And they also infiltrate into the heart, and toll-like receptor 4 activation is one of the main pathways involved. And this is why in our cell culture studies, we looked at LPS, which is a ligand for toll-like receptor. So you're activating this inflammatory pathway in the macrophages, and we saw that our lipoaspirate nanoparticles were able to lower all these inflammatory cytokines also to a similar extent as the cell culture-derived ones that is not shown here. And then we injected our human lipoaspirate vesicles into this mouse model, and we actually saw drastic results that we weren't expecting. So the inflammation in the heart was substantially reduced in the mice that received these human adipose vesicles, and TLR4 expression went down, which is the pathway that is involved in myocarditis. And we also saw that markers of macrophages infiltrating into the heart were reduced in the mice that received the human lipoaspirate vesicles. And we also saw that some complement proteins and receptors were involved in this. So we're looking further into the mechanism by which this human lipoaspirate vesicles can suppress inflammation in the heart. And so to conclude this part of the presentation, we asked, can we tap into the body's text message system to improve healthcare? And yes, we can. The diagnostic applications we're trying to develop is extracellular vesicle glycomes for brain metastasis, and the therapeutic applications are these adipose-derived extracellular vesicles for myocarditis, but also other inflammatory conditions. And with that, I want to thank my lab members and our funding sources and our collaborators. I want to thank you for your attention. I know you're all very busy, so thank you for coming to listen. Come and talk to me. Mayo Clinic in Florida is 5 minutes from the beach. And as you can see, I go there a lot. So we're always looking for people who want to change the world. Add me on LinkedIn, and you can follow our scientific updates on Twitter at Wolfram Joy. And this is my lab logo adapted from Johnny Walker. Thank you.
Anyone have any questions? You got about 10 minutes or so for questions.
So how do you make your disk nanoparticles? What's the synthetic process?
Excellent question. So it's photolithography and electrochemical etching of silicon wafers.
Chloroquine results are really interesting. So, clearance of extracellular vesicles is probably going to be a major factor in translating to therapy. And I'm sure you've thought about this, but what's the progress on combining the chloroquine with the exercise?
Yeah. That's a great question. So if we could use chloroquine not only for synthetic nanoparticles but for extracellular vesicles, yeah, I think it might work. But an alternative hypothesis is that perhaps the macrophages are the sorting facility, so that the vesicles will go there, but then the cargo will get resorted. And if this is the case, which we don't know at all, then it wouldn't be beneficial, but definitely something to look into. So we still don't understand. Most vesicles do go to the liver, over 90%, and this happens within a few minutes. So that's something definitely we need to look into.
Do the nanodiscs naturally concentrate in the lungs of healthy mice, or are these only mice that are cancerous lungs?
Yeah, that's a really good question. So actually, some of the smallest capillaries in our body are in the lungs. So we do see a slight increase in healthy mice, but this increase is even more dramatic when these mice have tumors. So you have the fact that it's the lungs, so the discs have a natural tropism for the lungs, but if you combine that with a tumor, you get the double effects. Yes? Go ahead.
Okay, yeah. So that was a great talk. So I had a question in terms of, in the realm of extracellular vesicles, there are subpopulations, and so I know that within the field it's very confusing as with the size differentiation. That's what I did in my graduate career. So I was wondering if you've actually looked at sort of the current size distributions in terms of what's denoted as an exosome or microvesicle, and seeing if maybe the two subpopulations may interact differently in different systems with myocarditis or in brain cancer. Like, does one contribute more than the other? Because I can imagine with the different size distributions, you get the larger vesicles, they may be carrying the larger cargo that can fit inside. So I just wanted to.
Yeah, that's a really good question. So, for instance, like you mentioned, some vesicles actually carry mitochondria, so big organelles, and these are usually the bigger vesicles. But actually today, there's nothing to tell us what is an exosome from a microvesicle. We can't tell by size, so the definition is that an exosome comes from within the cell, the vesicular system, while a microvesicle buds from the cell membrane. And unless we capture this through microscopy when it's happening, we shouldn't really say it's either an exosome or a microvesicle, but for sure the biogenesis pathways will affect their role and function in the body. So we haven't specifically looked into that, but it's a very good question.
So you touched on site-specific delivery, which is obviously becoming very important and very popular in the nanomedical research field, and also in terms of metastasis. So I know that can be a very deadly aspect of cancer. Because you can treat it, radiate it, chemotherapy, get rid of it, and then it comes back maybe a year or so later, and they lose a limb or whatever. So is there a way with these discs that you're talking about that they can attach to cancer cells within the bloodstream instead of just picking up in the tissue?
That's a very interesting point, and I'm not sure that anyone has looked to attaching to cancer cells while they're flowing. In my understanding, it's a rare event to actually capture cancer cells in the blood. To actually capture the extracellular vesicles from the cancer cells is more common, but that's an interesting approach. We haven't done it, but you should do it.
roses or glycans. Are y'all also going to look at like old glycans as well?
Yes, so right now the field of EB glycomics is very, immature, so we don't know a lot. So we would like to look at which part of the glycome is beneficial or is bad for cancer spread and progression. So we're definitely open to looking at all aspects of the EB glycome. Well, thank you so much. Amazing questions. Thank you.
Thank you so much.
Thank you. Thank you.
Great job.
Thank you.
You had a lot of information in really
Yeah,
just
brr.
1 hour.
Yeah.
Perfect.
Thank
Have a
you.
good day.
Yeah,
Safe
you
trip
too.
back.
You too.
Hi.
Hi.
Nice to meet you. So we thought about putting lectins on your discs. Because
Oh,
different,
no.
well, the one reason is most cancer cells, colorectal cancer cells,
Yeah.
they express GALDAC.
Yeah.
And there's lectins that are